Thank you for being here. So next we have Sara Cuadros Orellana. Sara is full professor at the Catholic University of Maule in Chile, a founding member and director of the Education and Development Committee of the Chilean Society of Bioinformatics and co-founder of the Chilean Network of Microbial Ecology. Her work focuses in the microbial communities in poly-extreme environments such as salt flats and hot springs. Thank you very much for being here today, Sara.
Thank you, Alejandra, for the introduction. Thank you for the invitation to be here today. I am sharing my presentation right now. Please confirm that you can see that.
Yes, perfect.
Okay, fantastic. So thank you again for the invitation, for being in this awesome event. I'm very happy to present some of the newest research we've been doing or conducting here in Chile. I'm originally from Brazil, and I'm glad to be representing actually both countries in this meeting. Well, the research we are conducting here now is about extreme environments. So we are using some metagenomic approaches to study the complexity of microbial communities in some extreme environments in the Andes. So the terrestrial hydrothermal systems, they are very complex systems. They of course are related to volcanic activity, and so they are naturally heated solutions of water. The hydrothermal environments, they are aquatic environments heated by this magma. But they also are very complex because of the different composition of the minerals that surround these volcanic systems. Well, the Andes are a very active volcanic system. It's part of the volcanic belt of the Pacific, as you can see in the upper right picture. And the Chilean territory comprises a huge part of this volcanic arc, and it's well, not so well studied compared to other thermal environments in the world. One very iconic environment that we know is Yellowstone Park in the United States. It's also a hydrothermal system. It's one of the most studied environments to date. But in South America, we don't know much about the complexity and the diversity of the microbial communities in these systems. So our objective is not only to learn about the diversity, but also to understand the main factors that are driving this adaptation and evolution of these microbial communities. So we are taking a more systemic view to these systems involving geology, also microbiology and bioinformatics, of course. So the mixing of fluids in these systems makes the composition of the waters to be different, right? And so even though they are in the same volcanic arc, each hot spring is unique. So this is the main rationale of our project, comparing different hot springs and fumaroles across the Andean arc. So we are focused in two areas of the Andean chain, which is the north of Chile and the central part of Chile. So here, just so you see a little bit of what these hydrothermal systems look like, I brought some videos to show you. So the first one is a fumarole. So you can see how the gases are released from the earth. So it's not aquatic completely. So in the below surface, you can find some more humidity, but actually what comes to the surface is mainly gases. And the sulfuric so the sulfur composition of these gases, you can see the crystals of sulfur salts accumulated in the surface. So these environments are quite acidic as well. So not only hot, the temperature of these vapors and gases is higher than 90 degrees Celsius, but also the acidity is another extreme factor. And to the right, you can see some mud pot. It's like a bubbling pond of mud. And in these environments, even though we believe this is too extreme for life to occur, there are some organisms that don't live without these conditions. They prefer and they need to be actually under these conditions to grow. So this is the main challenging aspect of the research. So here I show you two more hot springs, which are quite different because they are more to their surface. The water flows across a long area, and this makes an interesting scenario because it's not only the surface. The water is coming to the surface at high temperature, usually over 70 degrees Celsius, but it's flowing, and then you can find a gradient of temperature across this flow. And across this gradient, you can see also different types of communities developing. So it's a very good natural lab to study how the microorganisms adapt to different very small differences in temperature, in the availability of nutrients. You see here there are some vegetation around as compared to the ones that live only in mineral surfaces. So here's another one near a river in the region. So there's a complete different setup this is the diversity of hot springs that we find in the Andes. And all these four environments I just showed have never been studied with regard to the microbiology or the microbial community assembly. So we have done previous studies in Brazil when I was there. I've been in Chile for 4 years now. And we have studied other extreme environments. In that case, it was an acid mine drainage. And we have used metagenomic before to study them. So our idea was to apply the same methods to study the hot springs in the Andes. In the previous project, we managed to assemble many metagenomes, genomes out of the metagenomic data, so the MAGs, 244 MAGs. We also did single cell genomic, and we also cultivated some strains. So it was a very like a diverse approach. We tried different combinations of approaches to try to understand a previously unknown environment. So we are trying to do the same in here. So these are some publications that we got from the previous projects. So why to study hydrothermal environments? Well, one, because of biotechnology, we know that there's a lot of interest in the extremozymes, the thermophilic enzymes, also some bioprocesses. We have the bioleaching, which is a main process based on extreme microorganisms that can use energy from the minerals and make the conversions that the industry needs in the mining activity, but also for the evolution and the astrobiology. We know that the origins of life are probably connected to the thermophilic lifestyle. And in the sense of astrobiology, we know that this is maybe the clue for finding some signs of life and understanding possible life in other planets. And this is a big reason why to study this type of environment. So the main drivers of the diversity and the complexity in these systems is temperature. Definitely, we can see that the diversity follows the gradient of temperature, but also pH and chemical composition, and also light. So it's a very complex system. But we have chosen the latest project we have is to study a system in the highlands of Chile, in the Altiplano in Chile. This is called Lirima. This is a hydrothermal system that has been studied by my colleague Marta Hengst for the past 10 years or so. And it's a very interesting system in which you can see very diverse communities side by side, just because of the difference in temperature, because the water composition is the same as the ponds are connected, but they are connected, but one is shallow and the other is deep. So the temperatures are different, around 10 degrees difference between one and the other here in the left picture. So we chose this system to try to understand this complexity, considering that the main difference between them is the temperature. So we chose 3 ponds that are very near to are connected. The one that is has temperature 42, the red part I showed in the previous picture, has 42 degrees, and the blue greenish part is 53 degrees Celsius. So it's the main difference between them. All the other parameters are very similar. And a third pond, which is a little hotter, which is at 72 degrees. Just a reminder that this system is located at 4,000 meters above sea level. So when we talk about boiling temperature, we are not thinking of 100 degrees Celsius, but water will boil below 90 degrees. So the limits for life are not determined only by the temperature, but also by the altitude and the effects it has on the living systems and on the properties of biomolecules and other substances. Right, so water will boil at lower temperatures there. So we use this pipeline, which is a general one, very simple one actually, because we did the shotgun metagenomic analysis, which followed the traditional quality control, then the assembly. We could take a look at the taxonomic complexity of these reads directly without assembly by using some approach like Kaiju or Rust. MG Rust actually, and we did then the assembly because we wanted to recover genomes. Of course, we were interested in learning more about the organization of these genomes, so we did the assembly and functional notation, so to understand which genes and pathways they coded for, and some metabolic profiling, which I will show to you in a few seconds. Then we also took the other way after the assembly to try to recover full genomes out of the metagenomic reads. So we did the binning and then again the quality control and assignment of these bins, which are now called MAGs, which are the metagenomic assembled genomes. So this is what we did. Well, I will go very quickly through the details, right, of the quality control, but here are the tools we use, the type of reads we had. They were paired end reads with this setup, two ends of 150 bases. And the quality of the reads was very good. We lost very few, less than 5% of the reads after the quality control. We kept for the assembly both the ones that remained paired and the ones that didn't remain paired. So we included the unpaired reads to our assembly, which we did using Megaheat. So Megaheat is a very nice assembly tool for metagenomes because of its fast enough and very efficient as well. So it's been compared to other tools, and it has a very good performance. So using Megaheat, we could recover many contigs from all of these metagenomic sequences, even though the coverage was not so good compared to other studies we've done before. It was not shallow, but not very deep coverage. But even though we managed to recover many contigs with a very good length, right? So we can see here the maximum length of and the total length of the assemblies. So we were very happy with these results, and we proceeded with the annotations. So out of these metagenomic reads, we could recover information, functional information. So between 28,000 and 42,000 protein coding genes, which is a lot. We could really take a look at the differences between the metabolisms of these communities and try to understand this complexity using this information. So here I present a heat map. The heat map is presented, well, I cut some parts of the big map just to show some of the aspects, but they are organized like that. The three columns here are the LNS 3, 2, and 4. They are not quite ordered, but 2, remember, it's the 42 degrees. 3 to the left is the 53 degrees. So the coldest is in the middle one is to the left, and the hottest, sorry, is to the right, which is the LNS 4. So 42, 53, and to the right, the 72 degrees community. So this is the same for all the heat maps shown here. So we can see some differences in the metabolism. Some of the most evident ones is related to photosynthesis. We know that the limit for photosynthesis is around 70 or 75 degrees. At this altitude, we can see here that the completeness of the photosystem is not seen in the highest temperature, but there's almost a gradient, right? The highest abundance of photosystems is in the lowest temperature, and it decreases across the temperature gradient, right? And some other aspects, we found some features that are very ancient, of ancient metabolism, like the reverse Krebs cycle, which is shown here in the left panel, which appears only at the hottest temperature. So there are very specific interesting aspects of the metabolism. But other than that, we also took a look at the taxonomic diversity of these communities. Well, what the general scenario is that we have mainly bacteria. So this is to the left, you can see the cellular organisms, how they occur, their relative abundance. So 99% of the cellular organisms are bacteria in the coldest pond, so the 42 degrees Celsius, and only 1% are archaea. But this situation changes when we go to the 72 degrees pond, where archaea is already represented by 16% of the reads. So, and this situation probably follows at higher temperatures, we could find more archaea. This has been observed in other hot springs as well. Well, we do see this here, and we also can see the main phyla that occur in these environments. So basically, they are the most abundant phyla. They are consistently present in all the three ponds, right? Even though they vary a little bit in abundance. But what we can see actually here, a trend for the increasing the TAC archaea. The TAC archaea shows an increasing pattern of occurrence across the gradient of temperature. So this is the main observation that we see. And the TAC archaea still has some representatives that are only known by their 16S sequences and have no sequence genomes available. So the idea of taking a look at this diversity is actually to find the novelty, to find more information about taxa that are not well known to date. So here I presented the taxonomic distribution a little in a different view at genus level for some of the three ponds. So what we can see here, I tried to highlight the main observations to make it easier to present. In the coldest pond, which is LNS2, we can see mainly the bacteria from the Thermacia family. So the red star is highlighting this taxa, right? And also in the LNS3. So bacteria from Thermacia are abundant in the two coldest ponds. But when we go to LNS4, which is hotter than the others, we can see that even though they are still present, they now share the community. We can see some other taxa that are appearing, which belong to the archaea, as I told you before. So we can see Sulfolobus, we can see Pyrobaculum and Aeropirum, for instance. And here to the bottom with lower abundance, Thermoproteus, Acidobacillus, Aciditiobacillus. And also some other major difference, not related to taxonomy, but to the lifestyle. We can see the photosynthetic organisms highlighted, with this green rectangle, which are definitely more abundant in the two coldest environments, and they completely leave the scenario of the most abundant taxa at LNS4. So, what appears is a different group of organisms that are based on chemolithotrophy. So the energy source for the until 55 degrees or 60 degrees is photosynthesis. But after when we increase temperature, the source of energy is actually mineral, and the chemolithotrophic communities are the basis of all the ecosystem. And all the other taxa, they are syntrophic. They depend on the chemolithotrophic to obtain energy, right, and their resources. So there's a complete change from the market change we see is from LNS3 to LNS4. Well, some studies have been done before this one, but they were based on 16S sequences. They took a look at the microbial structure based on the sequencing of amplicons, and they could see a very similar scenario in the same ponds, the exact same site some years ago. But this was the first time that we could do the shotgun sequencing of these communities. And what was new from these regarding the taxa that we observed, and this is something we observed in our previous studies as well, is that even though amplicon sequencing goes very deep into the study of rare taxa, so we can see the structure of the community, and we can detect even the taxa that are present in very, very low abundance. In the case of shotgun, we don't see the very, very low abundance taxa, but we can see the taxa that are not seen by the amplicon approach, because the primers, there's a bias in the primers that we use that cannot detect all the groups. So when we do shotgun sequencing, we do detect some taxa that are candidatus taxa. Some are more abundant, like the one shown here, Bati archaeota, occurs at 8%. It represents 8% of the archaea in LNS4, for instance, but it wasn't seen previously by the 16S approach, and we can see it now and detect its abundance and try to study their genomes. And the same for other groups like the hydrotherma archaeota, which is a clade that is very like we know very few about, and it's detectable by the shotgun approach in these environments. Well, we then went past to the binning. I know I have little time left, so I will try to move a little faster in this. We did the binning approach using different tools, basically MaxBin, Metacarvo, and Binacle. Well, this work was supported by Claudio Quevedo, who is a student at Talca, University of Talca. We managed to recover 28 MAGs, 28 almost complete genomes right? Some are very high quality, so 13 of them are extremely high quality. This means they are more than 90% complete. They have lower than 5% contamination, so they are almost complete genomes or draft genomes. And these genomes are representing different interesting taxa, like the thermodesulfitimonas. This is a group of organisms that was described for the first time in 2017 in Russia. And there's very few knowledge about their lifestyle. So by adding 3 mags or 3 new genomes reconstructed from metagenomes, we will be able to contribute to the understanding of this group. But basically they have a very wide temperature tolerance, and they have a metabolism based on sulfur reduction, right? And we have a very good reconstruction of these 3 mags. Well, some other genomes, thermos, which is a caldoactive bacteria, we managed to recover 3 genomes. Allochromatium, which is a photosynthetic sulfur oxidizing bacterium, we managed to recover 2 genomes. Tepidemionas, also 2 genomes, and two very interesting archaea for which very few genomes, I would say 3 and 4 genomes, are available for these organisms. And we managed to reconstruct novel genomes from our metagenomic sequences. So we tried to locate these new archaea in the tree of life. Of course, we can do this using the 16S sequences. We can also do the whole genome comparison. And by doing this, we can see that other people have worked in similar studies. One of our colleagues in Chile, Leticia Barrientos, recently this year, she published the finding of 2 new mags from El Tatio. El Tatio is another hydrothermal system in the north of Chile, not connected to Lirima, but also to the north. And she recovered 1 mag also belonging to one of our mags. So I'm going back here to show Thermoproteus is one of our recovered genomes, and they also describe 1 mag belonging to Thermoproteus which could represent a new species of this genus. So we are very interested in comparing these sequences to see how they how similar they are, or if we can contribute to this description of a new species. So we also, of course, took a look at the viral fraction. We looked into the genomes of the reconstructed mags and also in the metagenomes, the abundance of viral defense systems in these organisms. We know very little how the viral how the viral diversity in hot springs will affect the adaptation and the evolution of the cellular organisms. So we are also interested in that. So I'm almost finished with the talk. What's next? Well, we will also study some other hot springs. So the one I showed to you in this study is near Iquique. So it's around this the uppermost green rectangle in the map shown here. But my institution is located in Talca, which is in the middle of Chile. And we are now studying other hot springs near Talca. And these hot springs are actually the videos I showed you in the beginning. They belong to this region, the Talca region. And the conditions are very diverse. pH from 2 to 8, temperatures from 30 to 94.5 degrees. And we are very excited to do this under with a support from ANID in Chile, which is a project that started last year and will continue until 2024. We will be very happy to collaborate with anyone interested in this type of ecosystem as well. We have been going to the field to collect samples. We have been sharing samples also with some colleagues interested in other aspects of the ecology of these organisms. So if you are interested in collaborating, please write to me and we will be glad to collaborate. So these are some pictures of the last sampling campaign. the reason why these systems are so poorly studied is because they are not so accessible. Actually, I must say that we're not even a big car or truck we could reach these sites. We needed to travel by horse, and it was a very long expedition. But fortunately, we found these new hot springs that have never been studied, and that we can take the first look ever to their secrets, right? So we are very excited to do that. We count on some collaborations, of course. This is my acknowledgement slides. I'd like to thank Mabel Vasquez, who went with me to field work, also some students of ours at UCM, some collaborators, Claudio Quevedo, of course, from UTALCA, who have been helping me in the bioinformatics aspects, and also some colleagues, Marta Hengst, who is collaborating with me directly, and some other colleagues from University of Antofagasta, Cristina Dorador, Jenny Blame from Fundación Biociencia, and Joanna Eisler, who is a virus specialist at University of Valparaiso. So these are the people I want to thank. And just one final invitation, please connect to RICHEM. RICHEM is the Microbial Ecology Network of Chile. It was formed by 17 amazing women, so it's also a female network. It's not only directed to women, but it was born by the efforts of 17 women. I'm very glad to be part of it. So we are on Twitter, and please connect to us. Okay, and also the Chilean Society for Bioinformatics, which was formed this year. So we are also on Twitter, and the Microbial Community, Microbial Society of Chile. So with that, I want to thank you very much for your time. I leave my email if you need to ask questions, or if you want to connect, and my Twitter user, so you can also be in touch. So thank you very much for your time, and I'd be happy to take questions if we have time.
Thank you very much, Sara, for such a great show and great talk. I am Cristina Marino, the chair of the next session, so I'm thanking you now. We have not a lot of time, but we have several questions in the chat and YouTube. If you allow me, I will read it for you.
Okay, sure.
So we have many people saying that it's an amazing show.
Thank you very much. So glad to hear that.
Yes, and thank you very much also for the invitation, for the talk, and for the invitations you've done for us. So one of the questions is if your results are published. I'm very interested in reading further about them.
Yeah, and actually not. We are preparing them for publication, but this is the at first hand the results. Yeah, we are still analyzing data. Yeah.
And this girl said that you say that more than one binning approach was used. Is there one you consider the best?
Actually, we have not come to a conclusion yet. Actually, each one had a pro and con. Binacle is a good one because it combines metacarval and also the bow tie calculation for the coverage, which is very important so we can be sure of the binning, because of course, the GC content is one of the main hallmarks of genomes, but we know that prokaryotes make horizontal gene transfer. This makes them to have some regions with bias in GC. So GC is not the only aspect to consider when binning. And so the coverage of the contigs of the reads is the main one, because of course, if a genome is present at a certain abundance, all the pieces of this contig should be present at the same abundance. So yeah, actually, Max Bean does that, but Binacle does this plus the scaffolding step, which is metacarval integrated. So I would tend to say that Binnacle is the good one, but even Binnacle has some flaws. So we needed to use VisBin, which is a fourth tool, to curate it manually. VisBin allows us to map and plot and see contig by contig and try to understand if it's contributing or not to the assembly. So yeah, I would be happy to be in touch through email to go deeper in the answer. But yeah, we haven't come to a conclusion.
We have another question. Did you have any retrospective data about habitat temperature in order to know if global warming could affect the community diversity?
Well, no, I don't have any background data, but global warming, I think that this is something that could really affect any type of microbial community, right? Actually, the imbalances in all ecosystems can occur. Not really related to the hot springs. We can see there the trends that are related to adaptation from very long geological times and biological times, right? We are talking about environments that are very similar to the origins of life, to the ones that where life originated. So in this case, we can see what is ancient and what is conserved, and also the differences that occur based on the interactions, right? Because some other organisms come to scene and they interact, and then the complexity increases over time. So the evolutionary aspect of these communities is something that we are actually interested, but not we haven't considered in this type of study the effects that could be caused by some event like global warming, which is more recent, right?
Yeah, true. So Sara, we have to thank you very much for the talk. There are other questions that we invite you to answer them through the Q&A system, because there are many other questions, but we're running out of time. So thank you very much for being here.
Thank you, Christina.
That's it.
And I will connect to YouTube, right, to see the questions and try to
in the
answer
Q&A you will see several questions that you can answer them now if you want.
Yeah, I will do that. Thank you.
Okay, thank you very much.